First off, we will define the two definitions for this concept, genetic diversity and inbreeding. Genetic diversity refers to the total number of genetic characteristics in the genetic makeup of a particular species, whereas inbreeding refers to the mating of closely related individuals, which tends to increase the number of individuals that are homozygous for a trait, and therefore increases the appearance of recessive traits within a population. Inbreeding has a negative impact on genetic diversity, but before we discuss this, we are going to look into how exactly genetic diversity occurs. Chromosomes and what they are comprised of is the key building block as to how large a species' gene pool is. Chromosomes are made up of DNA wrapped around proteins called histones. This DNA is comprised of long chains of base pairs, and the unique sequences of such bases are what code for the genetic makeup of an individual. It is the alteration of such base sequences through random mutations and recombination that variation within species occurs. Through processes of transcription and translation that are constantly occurring throughout the body, mRNA chains are created which code for particular amino acids that join together to form certain proteins uniquely associated with certain traits. It is these alternative traits of a gene that are recognized as the allele variations of a single gene. For example, one variation of a gene for coat color would be comprised of base sequences that code for a solid colored coat, while an alternative allele will code for the expression of a spotted coat. As a result of the different allele combinations, phenotypes of individuals differ, with heterozygous and homozygous dominant individuals expressing a solid colored coat, while the homozygous recessive individuals appear as spotted. Such variations of allele combinations occur during meiotic events, when homologous chromosomes line up along a metaphase plate and swap parts of their chromatid arms to result in 4 haploid chromosomes with new gene combinations. As a result, offspring differ from one another, so therefore, different and new combinations of alleles are what make up the variation within a species, and are crucial for maintaining diversity. However, inbreeding interferes in this process, as when individuals' genetic makeups are too similar or identical, even when the swapping of chromatid arms occurs during meiosis, no new allele combinations result, with identical haploid chromosomes resulting instead. So, when closely related individuals with desirable traits are bred together, offspring are almost certain to possess recessive traits, yet this is detrimental to crucial genetic variation being maintained within a species. Therefore, inbreeding is majorly detrimental, as through the repeated use of sires, breeding for specific phenotypes, breed barriers and line breeding, a smaller and more uniform gene pool results, which increases the percentage of genetic flaws and inheritance of recessive disorders due to occurrence of homologous traits that cause disease and exaggerated characteristics until they are detrimental to the progeny. Clearly, inbreeding is a negative practice for the evolution and survival of a species, so why is it performed then? Pedigree dogs are those pure bloodlines that conform to the breed guidelines and standards recognized across the world. Since the 19th century, when dog showing increased in popularity, breeding for conformation and color has become more prevalent. People have unknowingly reduced genetic diversity through inbreeding and overuse of popular sires. The distinctive characteristics and appearance in pedigree breeds that are popular amongst the general population all have some degree of inbreeding within their ancestral lines, and are therefore predisposed to certain conditions and diseases. One particular family of dog whose health has been negatively affected by inbreeding is brachycephalic dogs. Such dogs have an abnormally shortened head, upper jaw, and nose, and are exposed to a range of conditions such as eye problems, breathing issues, pyodermia, hip dysplasia, patellar luxation, and more. Some of the breeds affected are Boston Terriers, English Bulldogs, Pugs, Shar-Peis, and French Bulldogs. Brachycephalic airway obstruction syndrome are the respiratory problems that coincide with brachycephaly. These traits make it hard for dogs to receive oxygen into their airways. BAOS leads to mouth breathing, snoring, respiratory distress from struggling to breathe, and can lead to death. These dogs are not able to exercise due to breathing difficulties and are extremely prone to heat stroke and have disrupted sleep. Brachycephalic ocular disease is a syndrome that also occurs in such dogs, whereby they experience lesions of the eyelid, conjunctiva, and cornea. Dogs with this syndrome can exhibit abnormal protrusion of the eyes, an excessively wide opening of the eyelids, and an inability to close the eyelids completely. The abnormal head shape can also lead to lower medial entropion, dystichiasis, poor and excessive tear production, eye prolapse, and more. Some signs associated with these problems can include excessive blinking and sensitivity to light, discharge from the eyes and inflammation, and many of these symptoms can lead to partial or even complete blindness in dogs. Now let's look at a case study. Maggie is an 8-year-old English Bulldog and has had a history of noisy breathing. After many vet visits, Maggie was diagnosed. Maggie was diagnosed with having an inverted laryngeal saccules, an elongated soft palate, and stenotic nares. Maggie's surgery consisted of trimming of her elongated soft palate, widening of her nares, and removal of tissue from her nasal ring, as well as much more. But after the surgery, was the problem really solved? Despite after swelling from the surgery going down and Maggie being able to breathe, these surgeries occur daily and are invasive, expensive, painful, and increasing in prevalence.